Today we are going to talk about another metabolism of carbohydrate, that is gluconeogenesis. So first of all, let us talk about what is gluconeogenesis. The gluconeogenesis is the formation of the glucose from the non-carbohydrate source. You have to know this very well. So if there is formation of the glucose from the any non-carbohydrate source, then that is known as the gluconeogenesis. So the gluconeogenesis occurs especially in the liver. So the gluconeogenesis, the main purpose of this gluconeogenesis cycle is to maintain the euglycemia even in the fasting stage. So in the fasting stage, if we are not taking any carbohydrate, then the body by the help of gluconeogenesis maintain the normal level of the glucose by the gluconeogenesis. So the enzymes are found in mainly kidney and intestinal epithelium. The deficiency of the key gluconeogenesis enzyme causes hypoglycemia. You have to know this very well. And the muscles lack the enzyme glucose 6 phosphate. So the muscle cannot participate in the gluconeogenesis due to lack of glucose 6 phosphate enzyme. So the muscle lacks glucose 6 phosphatase, so that the glucose 6 phosphate at the end cannot be converted into glucose. So the gluconeogenesis cannot occur in the muscle. You have to know that the odd chain fatty acid yield one proponyl coenzyme during the metabolism, which can enter the TCA cycle, is the succinyl coenzyme and can undergo gluconeogenesis and can serve as the glucose source. But if you look at even chain fatty acid, it cannot do gluconeogenesis. The even chain fatty acid cannot produce new glucose because it cannot yield the gluconeogenesis substrate. The even chain fatty acid can only in acetyl coenzyme equivalent. So there is no formation of the new glucose by the even chain fatty acid. So this is the main concept. You have to know that gluconeogenesis occur in the liver and it maintains the euglycemia in the fasting stage. uh, it can also, the enzymes of the gluconeogenesis can also be found in kidney and epithelium. The muscle, however, the muscle doesn't participate in the gluconeogenesis as the muscle lacks the enzyme glucose 6 phosphatase, and odd chain fatty acid can produce new glucose, but even chain fatty acid cannot produce new glucose, cannot participate in the gluconeogenesis. Now let us talk about the gluconeogenesis from the pyruvate. Let us talk about the um, detailed cycle of the gluconeogenesis. So if you look in the mitochondria, if you look in the mitochondria from the pyruvate, by the help of the enzyme um, pyruvate carboxylase, the oxaloacetate is formed, and the oxaloacetate is impermeable to the mitochondrial membrane. So the oxaloacetate is converted to malate by the enzyme malate dehydrogenase, and here is the production of the NAD. So the malate is permeable to the mitochondrial membrane. As it converts to mitochondrial membrane, it comes out of the mitochondria in the cytosol and the further reaction occur. The malate is converted to oxaloacetate again by the enzyme malate dehydrogenase by yielding NAD+. And the oxaloacetate is converted to phosphoenol pyruvate by the enzyme phosphoenol pyruvate carboxykinase. This is the reaction you have to understand. And the phosphoenol pyruvate is converted to 2-phosphoglycerate, then it is again converted to 3-phosphoglycerate, and it is again converted to 1,3 bis-phosphoglycerate. So 1,3 bis-phosphoglycerate is again converted into glyceraldehyde 3-phosphate. So from the glyceraldehyde 3-phosphate, it can convert it to dihydroxyacetone phosphate, and also fructose 1,6 bisphosphate. And from the dihydroacetone phosphate also the fructose 1,6 bisphosphate can be synthesized. So 1,6 bisphosphate is converted into fructose 6 phosphate with the help of enzyme fructose 1,6 bisphosphatase. So this is the rate limiting enzyme. You have to know this very well. And the fructose 6 phosphate is converted to glucose 6 phosphate, and glucose 6 phosphate is converted to glucose with the help of enzyme glucose 6 phosphatase. This last reaction basically occurs in endoplasmic reticulum. So this is the main thing you have to know. So the again I am going to revise it. The pyruvate in the mitochondria is converted to oxaloacetate by the help of enzyme pyruvate carboxylase, and oxaloacetate is impermeable to mitochondria, so it is converted to malate with the help of enzyme malate dehydrogenase. The malate is transferred to the cytosol, and malate is again converted to oxaloacetate by the help of enzyme malate dehydrogenase. The oxaloacetate is converted to phosphoenol pyruvate with the help of enzyme phosphoenol pyruvate carboxykinase. And phosphoenol pyruvate is converted to 2-phosphoglycerate. Then it is again converted to 3-phosphoglycerate. Then it is again converted to 1,3 bisphosphoglycerate. And again 1,3 bisphosphoglycerate is converted to glyceraldehyde 3-phosphate. The glyceraldehyde 3-phosphate is converted to fructose 1,6 bisphosphate, or it can also be converted to dihydroxyacetone phosphate. After converting to fructose 1,6 phosphate from the glyceraldehyde 3-phosphate or dihydroxyacetone phosphate, the fructose 1,6 bisphosphate is converted to fructose 6 phosphate with the help of enzyme fructose 1,6 bisphosphate, which is the rate limiting enzyme. Fructose 6 phosphate is converted to glucose 6 phosphate, and glucose 6 phosphate is converted to glucose with the help of enzyme glucose 6 phosphatase. The conversion of the glucose 6 phosphate to glucose occurs in the endoplasmic reticulum. So this is how the glucose is formed from the pyruvate. So now let us discuss about the regulation of the gluconeogenesis from the pyruvate. So basically you all know that the regulation always occurs in the irreversible enzymes. So the enzymes which does the irreversible reaction, mainly there the regulation of the enzymes occur. So in the mitochondria, for the proper functioning of the pyruvate carboxylase, we need biotin, ATP and acetyl coenzyme. So without biotin, ATP and acetyl coenzyme, the pyruvate carboxylase doesn't function properly. And the phosphoenol pyruvate carboxykinase requires GTP for the proper functioning. So phosphoenol pyruvate carboxykinase requires the GTP. And another is in the rate limiting enzyme. The rate limiting enzyme is also regulated. The fructose 1,6 bisphosphatase is regulated. Mainly the AMP and fructose 2,6 bisphosphate inhibit the enzyme fructose 1,6 bisphosphatase, whereas the citrate activate the enzyme fructose 1,6 bisphosphatase. So this is the main regulatory steps you should know. This is the main thing you have to know in the gluconeogenesis from the pyruvate. So these are the various steps. I have briefly discussed about the initial introduction of the gluconeogenesis that mainly occurs in the liver. Later on, I discuss about the detailed cycle, I mean the detailed reaction of the gluconeogenesis from the pyruvate, and I also discussed about the regulation of the gluconeogenesis from the pyruvate. The regulation mainly occurs in the irreversible enzymes. So these are the main things you have to know in the gluconeogenesis from the pyruvate. I hope you understood this lecture. Thank you.